Good evening, ladies and gentlemen. It's a pleasure to come up to Edinburgh this evening to present the Silence Therapeutics story. So a little bit of a change in gear. Silence Therapeutics is a biotech. We're based down in London, but we have research facilities in Berlin. And the usual disclaimer. So I'm going to start a little bit in kind of basics of biology. And I think there's been a couple of life sciences talks this evening, so I hope I'm not repeating myself. But just so everyone's on the same page, so think about kind of your genome, your DNA, the blueprint of all your, what makes you who you are. The DNA is inside the cell, and ultimately it gets made into protein, and protein are kind of the workhorses in your body. Hemoglobin is in your blood, it's spherical, and it carries oxygen around your blood. Now, there's a middleman in between the DNA and the protein, and that's called messenger RNA. And when you get a disease state where you have a genetic error in the DNA, the message is in the DNA. And if that gets translated into protein, you get faulty protein, and I'm talking very simplistically now, but that's what causes diseases in many cases. And this messenger RNA is kind of culpable in transmitting this faulty message. Now, this is where Silence Therapeutics comes in. So we have developed a proprietary technology called Rna Interference. This was developed about 20 years ago, won the Nobel Prize in 2006, scientists in the US. And there's a handful of companies that have this technology, and we're one of the pioneers in the space. And what we do is we engineer these short oligonucleotide drugs that are perfectly complementary to the target that we're seeking to go after. And when you inject the drug into the body and it binds very, very specifically to this messenger protein, this messenger RNA, what happens then is it gets degraded. So what you're doing is you're very precisely, we say, knocking down the target that you're trying to go after. And thereby, in this case, in this simple example, the faulty protein no longer gets made. So that's what we do. We have over 18 years experience in this. As I said, over 30 scientists in Berlin, real experts in this space, about 50 employees across 2 sites, and we're worth about $400 million today. We're just about to go into the clinic with our lead program, and I'll tell you more about that over the course of the presentation. We are a public company listed on AIM, and as of the end of December, we had $44 million in cash, taking us into the second half of 2021. Now, Silence is a platform to product play. So what does that mean? So I talked a little bit about the technology, and I'll go back over it again in a few minutes. But essentially, we have a technology that you can translate into almost unlimited targets. So there's 7,000 proteins expressed in the liver, which is where the cell type we go after. And in theory, you could take our platform and you could go after any one of those targets. You can also think about personalized medicine. If, cancer genomes, for example, where everyone has a slightly different genotype, you could customize your siRNA in theory to go after each of those separate genotypes. So the platform is sound. We have freedom to operate, strong intellectual property position, and it's now about converting it into products. And I'm going to talk about 2 of those potential products this evening, but the point I'm trying to make is that we have many more ideas and the ability to translate the platform into many more products going forward. I am the CFO. I joined Silence about 3 years ago. I've got a PhD in biochemistry, and I somehow managed up training as a chartered accountant as well. So I don't know, I must have fallen and hit my head one day, but that's what happened. And I spent a number of years at GSK. I was the area finance director for one of their business units, and I became CFO of Silence early last year. We are currently without a CEO, but Ian Ross is interim executive chairman. Ian's got over 40 years experience in life sciences, and a great chair to lead us through this period. And I think very importantly, our head of R&D is Dr. Josh Campy, and Josh joined us last summer. And Josh has really kind of he's been down this road many times before. He was the chief medical officer of a company called Presenza, which IPO'd on NASDAQ, 9 times oversubscribed, massive deal with GSK, ultimately got bought by BioMarin for $680 million. But he's spent many years at Novartis and GSK and others and really knows the space very well. So I think a really strong team. So just a couple more minutes on the platform itself. So I mentioned earlier we have this oligonucleotide sequence. So big word, it's really kind of, it's the same thing that our DNA is made of. So it's really about complementarity to the DNA or the RNA that we're going after. This technology is, it's very reproducible in cells. So you can really go after anything you're looking at in cells. The difficulty with our industry has been about delivery. So it was about getting it inside the cell that you're looking to target in the body. And there was numerous clinical programs over the last 15 years or so that tried to do that and weren't quite successful, although one has been approved by the FDA in 2018. But what has happened in the last couple of years is that we figured out this beautiful little molecule here, which is called a GalNAc sugar. And by appending this simple sugar to our sequence, we can exquisitely target the drug to one particular cell type, and that is hepatocyte cells in the liver. And it is not, it's about 99% uptaken by that particular cell type, and the rest of it gets excreted. So this just shows you a cartoon of that happening. So we're talking about subcutaneous injections, maybe 1-2 mils of drug. It gets taken up in this one particular cell type in the liver, and then it gets into this intercellular machinery. And I won't go through the detail here, but it's really about targeting the DNA that we're trying to go after. Now, Silence had this technology in development probably since 2015, 2016. We would always say that our technology is competitive with the best in the world, but we didn't have any external validation to back that up until last summer. So last summer we did a large deal with Mallinckrodt Pharmaceuticals, where Mallinckrodt paid us $20 million up front in cash. They also made a $5 million equity investment. And the total deal size, it was worth potentially over $2 billion if everything plays out as we would like it to. It was a 3-target deal. One of those targets we had done a little bit of development work with. The other 2 are options, and no work has been done on those to date, but they do have options to take those forward. And I think off the back of this deal, we went from about $60 million in market cap up to, let's say 250 around Q3 last year. In January this year, we did a second deal with Takeda. The economics are much smaller, so single-digit millions of US dollars of research funding. But I think most people will agree that Takeda is a very strong brand. They do not do partnership deals with companies they haven't done a lot of diligence on, and they did that with Silence. And so really pleased to now partner with Takeda as well, and confident that we can do further deals along these lines going forward. So we also have 2 existing out licenses, and people ask about them a lot, so I'm just going to spend a minute on them here. We did a license agreement with Alnylam Pharmaceuticals, whereby we get 0.33 to 1% royalty on sales of a drug they sell in Europe called Onpattro. They sell it elsewhere as well. First royalties are expected to were received on that in Q4, and we'll be reporting on that going forward. These are not material amounts of royalties, though, I should say, when you have a royalty rate like that, and they had just launched the drug in 2019 in Europe. We also have a license agreement with Quark Pharmaceuticals, a royalty rate of about 0.5 to 4% on that. But I think more importantly, Novartis have an option on the 2 programs that Quark are running that we're invested in. And if Novartis take that option, we would receive 15% of anything that Quark take. Again, you're talking, potentially in the order of tens of millions of pounds of NPV. I think where we stand today at 400 million, perhaps not significantly moving the dial. I should say Quark is a private company, and we're not privy to any information on that. They did read out their phase 3 trial in early 2018, or at least they said it was finished. We haven't seen any of that data yet, so we're not very optimistic around Quark, but it is an upside nonetheless. This is our pipeline. So we've got 3 assets that are disclosed and we're talking about publicly. We do have a broader pipeline of assets behind these. And I'll spend a few minutes on the first couple in the time remaining. So SLN124 is for the treatment of iron overload disorders. So these patients, and we're talking about relatively rare diseases, beta thalassemia, myelodysplastic syndrome, in the order of about 60,000 patients in the US and EU for beta thalassemia, about 100,000 for myelodysplastic syndrome. They're anemic and they get regular blood transfusions. And when they get these transfusions, you see a huge kind of bolus of iron that comes alongside the transfusion, and the body cannot deal with that free iron, and that causes all sorts of issues. There are drugs out there that soak up the iron. They're called chelators, but there are some toxicity issues with chelators as well. And they don't work on secondary iron, so that kind of buildup of iron in different organs in the body that results from these diseases. So what we're aiming to do with our drug is to enhance erythropoiesis, so encourage more red blood cell production in the patients. That means they're less anemic, they have less red blood cell transfusions, and they have less iron buildup in their organs. So remembering back to the introduction, so what our technology does, so we knock down a target microRNA, messenger RNA. So the target in this case is something called TMPRSS6. And it's a little bit convoluted, but try and bear with me. So hepcidin is a hormone that regulates iron levels in the body. TMPRSS6 is a negative regulator of hepcidin. So TMPRSS6 naturally dampens down hepcidin in the body. What we do is we knock down TMPRSS6, and by doing that, negative times a negative, we actually increase the levels of hepcidin, and thereby reducing iron levels, improving erythropoiesis and reducing anemia. And I'll just show you one real piece of data on this, and this is an experiment in mice. So the mice are injected and they're followed for 6 weeks. And you can see when you look at the target, so TMPRSS6, after 1 week after the drug's administration, you can see that it goes from, this is normalized, so from 1 down to, let's say, 0.1. So about a 90% reduction in TMPRSS6. And then when you follow it over time, so over 3 weeks, it starts to climb back up, and after 6 weeks, it's starting to climb back up again. Importantly, though, what happens to iron levels? And hepcidin is missing from the middle part here, but hepcidin increases in line with TMPRSS6 reduction. And as that hepcidin increases, you see that the iron is reduced in these mice in this instance. So SLN124 is just about to enter the clinic. We have, I think, 7 sites across Europe. We've said that we're going to dose the first patient in Q1 this year, and aiming to have interim clinical readout the second half of this year on that program. The second program I want to tell you about this evening is called SLN360, and it's a cardiovascular disease program targeting something called Lp(a) or lipoprotein A. Now, this is something that was discovered many, many years ago, and it was kind of it was a little bit forgotten when, in the grand scheme of things with LDL and HDL and all the statins in development, etc. It's a relatively minor component of the lipids that are in your blood. It's less than 5% of the lipids. But more and more, it's being appreciated as a significant causative risk factor for cardiovascular disease. Now, in a population, when you look at a biological parameter, you normally see something like this distribution curve. So most people have something around the middle. Some people have less, some people have more. But when you look at Lp(a), you get this skewed curve whereby the vast majority of the population have low levels of Lp(a). but about 20% of the population have over 50 mg per deciliter of Lp(a). Now, what does that mean? When you actually translate that into cardiovascular risk, you can see that the risk of death, the risk of myocardial infarction, heart failure is significantly increased for people who have high Lp(a). And this is a huge population. So if we just take the USA and Europe alone, over 100 million people who have high levels of Lp(a). Now, the interesting thing about high Lp(a) is that it's genetically independent. So you can be slim, you can be overweight, you can do exercise, not do exercise. It doesn't matter. It doesn't matter. Whatever your Lp(a) level is at birth, it's going to be your Lp(a) level as you grow, and that risk is with you throughout your whole life. It's something that's easily measured. If you go to your GP and ask for it to be measured, they can organise it. It's not something that's standardly measured, though, and the reason that is because there's not a standard of care as we speak. Now, we are a fast follower in this space. So Novartis and a company called Axia are just about to start a phase 3 trial, 7,000 patients, 4 years. We are third in the race to develop a drug for Lp(a). But as you can see, an absolutely enormous market and a huge unmet medical need here. No standard of care as we speak. This just shows you some of the data, and so this is an experiment in an animal model where we dose it. There's 3 different dosing levels, but let's just look at, say, 3 mg per kg, the green line. You can see, and this is the control in red, you can see very rapidly, within 14 days, you get about a 90% reduction in Lp(a). And when you follow that out, and let's just look at some of the higher concentrations, you can see with the higher concentrations that actually it's barely detectable even 2 months later. So in theory, and as we hope, you could have a drug that could act for 2, 3, 4 months after a single administration. And we have seen a new heart medicine just conditionally approved by the FDA called inclisiran, which is a similar kind of drug to ours, but that lasts for 6 months. So you go to the doctor twice a year to have this injection. So in summary, on SLN 124, we are just about to enter the clinic. I should say we have not been in the clinic in a number of years. This is a really big moment for Silence, and it will be the first of many clinical programs, but a great milestone, with data to report in the second half of this year. I didn't say it, but on SLN 360, the cardiovascular drug, we're on track to file for the start of a clinical program the second half of this year. IND just denotes that it will be US filing. So the SLN 124 is a European program. This will be both a US and a European program. And we expect the first patient shortly thereafter. And I didn't talk about it at great length, but one of the targets that we're working with Mallinckrodt, we also hope to have that in the clinic in 2021. So by 2021, we would hope to have 3 clinical assets using our technology. We just announced yesterday morning that we have a scientific advisory board just set up. It's being chaired by Sir Gordon Duff, who's an eminent person in the field, and a really great person to lead us, and a great advisory board in general. Some of the names there are world class. We are deep in recruitment of a new CEO, and we do think over the next 2 to 4 months that we'll have that announcement ready. But as you can imagine, we're in no rush. We want to secure the right person, and that process is underway. I mentioned on the Takeda side and clearly kind of targeting further opportunities along that track. And then on the corporate side, we announced that we would form a US subsidiary in 2020. This is really around a direction of travel towards the US. I think at some stage we will need to raise significant amounts of money as we go into large phase 3 trials, and the US is a place where some of that capital can be found. But the US subsidiary allows us to kind of be a bit more focused on the ground in the US. And then just in summary, as I said, by 2021, 3 assets in the clinic, and investigating our medicine candidates in over 100 patients. Thank you for listening. Happy to have any questions.
So I think we've got one question here.
Hi, could I ask you a quick question on your royalty rates?
Uh-huh.
You seem to have quite a big spread between point something up to 15. What is the major determinant in determining what royalty rate you get?
Yeah, it's a good question. So if you out-license a piece of technology, if you out-license a single patent, for example, which is part of the value of a drug, but it might be one of 10, 15 patents, then the royalty will be low. If you out-license a whole drug, so the Mallinckrodt collaboration, we're out-licensing effectively, that whole drug in many respects, then the value that you can gain from that type of out-license is much greater. That's really and obviously it's around kind of the indication, the competition, etc. as well.
Thank you. And any other questions in the room? I had a quick question just in terms of how sort of your model might work going forward. Are you expecting to do a lot more kind of collaborations with larger pharmaceutical companies? Is that something you'd expect to see?
Yeah, so I think it's a mix. I mean, it's critical that we have our own proprietary platform and that we bring that forward as far as we possibly can. We'd love to take drugs all the way to the market ourselves.
Yeah.
Clearly, you need to fund that, and one of the ways to fund that is through non-dilutive funding, through partnerships. So it's always a mix of the two. You also need to think about the indications themselves. A rare disease like SLN124 is something that is well placed for us to develop the whole way. A lot of people say, well, SLN360 for cardiovascular disease, can a small biotech do, thousands of patient trials?
Yeah.
Perhaps not. So it's trying to marry it all up in that regard.
Yeah, and so just sort of in a bit more depth, how do you sort of balance the decision between sort of going down the road of a collaboration or sort of taking things forward yourself?
Yeah, so there's many factors. So you look at what you have in the pipeline, how far advanced they are. We don't want to partner SLN124 and SLN360 right at this moment because, well, we're just about to enter the clinic.
Yeah.
I think to give away your lead programs like that at this time is probably detrimental to value. So many factors that we'd consider.
Great. Any more questions in the room? In which case we'll leave it there. Thank you very much. Thanks for your time.